Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you love getting smarter faster, please hit that like and subscribe button and share this deep dive with your friends and family. It truly supports the Sand Science Experience. Today we are opening up one of biology's, greatest paradoxes. How does life even function when its essential operating system is split between two separate genetic manuals? I'm talking about the huge set of blueprints we call nuclear DNA, what you get from both parents. And then there's the tiny separate power plant instructions, the mitochondrial DNA, or mtDNA, which you only get from your mother. They have to work together perfectly to build you, but what happens when that cooperation is stressed or mismatched?
And that is the core of the whole Mitonuclear challenge. Our mission in this deep dive is to guide you through a truly monumental study using Drosophila melanogaster, the common fruit fly. The researchers here, they didn't just look at the two genomes, they tried to quantify the precise weight of every possible factor influencing fitness. That means measuring the effect of the nuclear genome, the mitochondrial genome, the environment, and critically, all the complex ways those factors interact. What geneticists call epistasis, or GXG, GXE, and even GXGXE, we're looking for what's really driving biological variation.
Okay, let's unpack this system first. The whole reason we have to care about this genetic partnership is it's all about energy production, right? Almost all cellular energy is generated by oxidative phosphorylation, or as we call it, OXPHOS.
Yeah, and that's pronounced OXPHOS.
OXPHOS, got it.
Right, and you can think of OXPHS as the engine assembly line in the cell. This line is made up of 5 enormous protein complexes, and the key parts, the actual physical subunits, are encoded by both the nuclear and the mitochondrial genomes. They have to fit together like hyper-specialized Lego blocks. If they don't, the whole engine just misfires.
And for this study, we're focusing primarily on complex 1, are we?
Yes, complex 1 is the largest and really the most critical starting point for that whole engine.
Right.
It's the primary bottleneck for energy flow, and because it needs parts from both genomes, it's incredibly sensitive to any genetic mismatch.
This dependency, it brings up a core evolutionary question. Is the variation we see in traits, like how fast a fly can fly or how big it grows, is it driven mainly by simple linear effects, A plus B equals C? Or is the real action hidden in the complex interactions, the epistasis?
And if it is epistasis, we should define it. GXG isn't just A plus B. It's when the effect of your mitochondrial genome, let's call it A, completely changes based on which nuclear genome B it's paired with.
Okay, so the researchers set out to map every conceivable pairing. They created 90 mitonuclear lines. That is just a massive undertaking.
It is. So what they did was they started by collecting 22 distinct mitochondrial DNA haplotypes. These included variants from within the common fruit fly population, so local variants from Beijing or Zimbabwe. But, and this is crucial, they also included highly divergent mtDNA from totally different species, D. simulans and D. yakuba.
So that's millions of years of evolutionary separation.
Millions, yeah.
So they were engineering both subtle, local disagreements and these massive cross-species mismatches.
Exactly. And they paired all 22 of those mtDNA types onto just 2 different standard nuclear blueprints. These 2 nuclear backgrounds, OreR and DGRP375, are basically reference strains that every fly lab uses.
So it allowed them to test if a specific mtDNA performs differently depending on its nuclear context.
Precisely. And then to really challenge the system, they introduced environmental stress.
Right, specifically targeting that crucial complex 1.
That's the one. They measured 4 traits that are extremely metabolically demanding: body weight, climbing speed.
That's negative geotaxis.
Negative geotaxis, yeah. Also flight performance and development time. And to simulate that stress, they cultured the flies on 2 diets, a standard one and one laced with rotenone.
And rotenone is that pesticide that directly inhibits complex 1. It's basically sabotaging the energy assembly line on purpose.
You got it.
So we have this massive data set, 90 lines, 2 environments. Now, given that the nuclear genome has, 20,000 genes, and the mitochondrial one has 13, it seems kind of obvious which one should be in charge. And overall, the nuclear DNA did dominate the variants, didn't it?
For most traits, yes. The nuclear genotype really sets the performance ceiling. It was the major driver. For example, for female flight performance, the nuclear background explained about 75% of the variation. It determines the potential. But here's where it gets really interesting and where the data just feels so counterintuitive. Body weight. Why would something as fundamental as the physical size of the fly be controlled more by the energy component than the structural blueprint?
This result is a huge surprise. For body weight, the mitochondrial DNA variation had a larger main effect than the nuclear DNA. For female flies, the mtDNA effect size was significantly larger.
Wow.
It suggests that for a basic metabolic trait like just sizing the organism, the efficiency of that tiny maternal energy system is just disproportionately influential.
So if your power source is weak, your capacity for growth is limited, no matter how good your blueprint is.
That's a great way to put it.
Let's just pause on that for a second. The power source is controlling the scale. And that really challenges how we usually think about genetics.
Absolutely. But if the nuclear genome is the major player overall, the biggest surprise comes when we look at the interactions. Let's talk about GXE, the interplay between genes and environment. This interaction, how the genes react differently to stress, that proved to be incredibly significant, didn't it? Especially for traits directly linked to metabolic output, like climbing and development.
And this is maybe the most earth-shattering finding in the whole paper.
Okay.
For development time, the mtDNA by environment interaction, the GXE effect.
Interaction, right.
Its effect size was actually larger than the effect of the Rotenone stress by itself.
Wait, hold on. Let me get sure I'm getting this right for everyone listening. You're saying that the stressor itself, the Rotenone, caused less overall variation across the whole population than the interaction between that stressor and the specific mitochondrial type of fly happened to have.
Precisely. It's a profound shift in focus. It says we should be less focused on the environmental stressor itself and more on identifying which individuals have the sensitive mitochondrial variants. The stress simply unmasked the genetic differences that were already there.
So this must put a big damper on the idea of pure genetic epistasis, the GXG mismatch. Did the actual pairing of the two genomes matter as much as the external stress?
Surprisingly, no. Mitonuclear epistasis, the GXG, was generally pretty limited. It was only strongly significant for one trait, which was male body weight, and maybe marginally for male climbing.
And that's a crucial finding.
It is. It suggests that genetic background mismatches alone are less frequent drivers of variation than the environment is. At least in this system, the environment is the great unmasker.
Okay, so these results, they must create a conflict with some established evolutionary theories, right?
A central conflict, yeah. For decades, the main idea was this compensatory coevolution model. It's pretty straightforward. The nuclear and mitochondrial genomes co-evolved to fit perfectly, so if you pair a divergent interspecies mtDNA with a common nuclear background, you should get a major fitness breakdown. Mismatch should equal failure.
Right. So mismatching a D melanogaster nucleus with, say, a D yakuba mitochondria, that should be catastrophic. The engine should just seize up.
But the study found the exact opposite, and this is the major paradox they uncovered. The highly divergent outgroup, mtDNA, the simulans and yakuba variants, they actually showed intermediate trait values. And even more surprising, they showed weaker interaction effects, both GXE and GXGXE, compared to the huge variations seen within the D melanogaster populations.
That is just fascinating. So the old co-evolved pairs were actually more stable and predictable, while the newly evolved transient variants within the species caused the most instability and reacted wildly to stress.
Exactly. It completely challenges that simplistic idea that any mismatch leads to dysfunction.
So if it's not adaptive coevolution driving divergence, what is it? What maintains the stability across species? And why is there so much instability within the species?
The results really point to a model of efficient purifying and stabilizing selection. It's just relentlessly acting on the mtDNA. The most disruptive variants, the ones that make the engine seize, they get purged very quickly before they can ever become fixed differences between species.
And how do we know they're the most disruptive? Is there molecular evidence?
There is. The researchers found that these mildly deleterious variants that are still floating around within the melanogaster population, the ones causing the chaos, they tend to be highly concentrated. They're enriched right at the physical protein contact sites in complex 1.
So right where the two parts have to meet.
Right where they meet. 62.5% of the amino acid mutations were clustered there. And they're often radical changes. The really bad variants just don't last long.
That makes perfect sense. Stability over millions of years means anything truly harmful gets deleted, and fast. Okay, we have to quickly touch on a very specific hypothesis. The mother's curse.
Ah, yes, the mother's curse.
The idea that male-harming mutations build up in mtDNA because selection only sees female fitness.
Right. They explicitly tested for it, looking for any sign of sexual antagonism, strong or weak form. And across all the complex traits they measured, they found zero evidence for mother's curse in this system. None.
So if we take this entire complex map of genetic interaction. How do we apply it to human health? What's the translational impact for us?
It has very direct implications for human medicine, specifically for mitochondrial replacement therapy, or MRT. When you're using a donor mtDNA to prevent transmitting mitochondrial disease, you have a really serious question about donor choice. If this fruit fly research is right, it suggests the mitochondrial variation found within a population is more likely to harbor these transient, mildly deleterious mutations.
The ones that cause the most instability under stress.
Exactly. Which implies that using donor mtDNA from highly divergent sources might actually be a safer choice.
So you're saying that if you need a donor part, maybe you shouldn't get one from your direct neighborhood, where people might share these slightly faulty versions. Maybe you should go for a highly robust, well-tested divergent version that's already proven its stability.
That's the implication. Greater polymorphism within a population might actually mean greater instability when you're exposed to stress. That's vital knowledge for MRT.
That's an amazing, actionable insight from watching fruit flies climb walls.
It is. But it also leaves us with a provocative final thought for you to consider. Stabilizing selection seems incredibly powerful in high population-sized species like Drosophila. It's rapidly purging these bad mtDNA mutations, so that raises a critical question. How might the accumulation of these mutations accelerate in species with much smaller effective population sizes? Species where that purifying selection is inherently weaker. If that cleansing mechanism slows down, the risk of mitonuclear incompatibility, and so your susceptibility to environmental stress, could go way up in those lineages.
A truly fascinating problem to leave us with, the interplay between population size and genomic vulnerability. Thank you for walking us through this incredible deep dive. And thank you to you, our listener, for joining us on the Sand Science Experience. Please like and subscribe, and we'll see you next time for more thrilling science.